last macromolecule group is the nucleic acids. So like all the others, we're going to go through the different parts. So the name given to the monomer of a nucleic acid is called a nucleotide. And a nucleotide has 3 different parts. So those 3 different parts are, it's going to have a 5-carbon monosaccharide. So here we can see a 5-carbon monosaccharide. We have 1, 2, 3, 4, 5 carbon molecules. And then, because it's a monosaccharide, you can see we have our hydroxyl groups and hydrogens filling in the rest of it. This is going to be the base of our molecule. The second component is called a nitrogenous base. This can be a somewhat complex molecule. You can see these in figure 320, but this is the nitrogenous base, and we can see the nitrogens throughout this molecule. Also, I should say this is in figure 320. So, we can see the base of our molecule. Remember, they're going to call this a sugar. Here's our nitrogenous base. We will attach it to one side in order to create our molecule. And so you can see that the 5-carbon monosaccharide is attached to a nitrogenous base. So there we have 2 parts of our nucleotide. I'm getting used to the camera, so sorry if I'm there. The third part, we talked about functional groups. We have these phosphate groups. And so here we have our phosphate molecule surrounded by 4 oxygens. And on another part of our molecule here, we will pop off a hydrogen and we will add our phosphate group. And so this all together gives us 1 nucleotide. Our 5-carbon, this is hard to do also, our 5-carbon monosaccharide, our phosphate group, and our nitrogenous base. 1 nucleotide. Now, one of the things you see here in our picture is, let me put my 5-carbon monosaccharide back together. So, 5-carbon monosaccharides, there are 2 different ones that we can have depending on what polymer we're going to make. You can either have ribose or you can have deoxyribose. These are both 5-carbon monosaccharides. For nitrogenous bases, this is just one of them. You can see the others in the figure 320. The only one they don't show there is uracil. So the nitrogenous bases come in 5 forms. They're called adenine, which will symbolize with an A. Cysteine will symbolize with a C. Thymine symbolized with a T. Guanine symbolized with a G. And uracil symbolized with a U. So that can change depending on the nucleotide you're looking at. The phosphate group is going to stay the same, however. So we can also build polymers from our nucleotides. And when we do, we will get different polymers. So when we do dehydration synthesis, we can create polymers of nucleic acids. And this is your molecule DNA or RNA. So DNA will have deoxyribose as its 5-carbon monosaccharide. RNA will have ribose as its 5-carbon monosaccharide. And then they are going to be held together by what's called a phosphodiester bond. So that is our 4th covalent bond that can hold these polymers together. So if we were to look at this on a bigger scale, so if we were to take a nucleotide plus another nucleotide, these would be my reactants, and I were to do synthesis, I could create a DNA molecule plus water. But this would have to be a nucleotide that had deoxyribose as its base. This would also have to be a nucleotide that has deoxyribose as its base. Or I can create an RNA molecule. If ribose is at the base of this one and ribose is at the base of this one, I could then create a ribonucleic acid. So those are the 2 major molecules that we can can build. And we can see this in figure 3.21. So figure 3.21, you can see that they have combined a guanine with a uracil with an adenine with a cysteine in order to create an RNA molecule. Now, some things we need to know about DNA and RNA. So, at some point in this semester, probably on the next test, I'm going to ask you to compare and contrast RNA and DNA. So therefore, we need to know some of the different characteristics. One of those is shape. So you can see in the figure 320, DNA is shaped as a helix, which is why you often hear it referred to as a double helix. So this means you have one strand of DNA here, and you have a second strand that spirals with it. So therefore, it is in a helical pattern. RNA is only one-stranded. So it is going to be a linear structure. You're just going to have a single strand. So that means another characteristic is number of strands that make up the molecule. This will have two. This will only have one. Here we can see our two interroping in a helix. This one is just a linear molecule and does not have a pair with it. Another characteristic is the monosaccharide. Here we have deoxyribose. Here we have ribose. And then a fourth characteristic is those nitrogenous bases. The monosaccharide, this tells the difference between DNA and RNA. Nitrogenous bases tells the difference between nucleotides. We can only put 4 different nucleotides on DNA. We can only put A, C, T, and G. In RNA, it also can only take 4 different. However, it can take A, C, and G, but instead of T, the uracil will now come into play. So this is why this is the one that tells the difference between the two molecules. Because if I have an A, and I'm just looking at that nitrogenous base, I wouldn't know if it's a DNA molecule or an RNA molecule because A can appear in both. So if I have DNA molecules, I'm not going to drop the molecule, I'm just going to write deoxyribose. Then it can have an adenine on it, and it would have a phosphate group over here. This would be the adenine DNA nucleotide. We could have cysteine on there, and that would be a DNA cysteine nucleotide. Or we could have guanine on there, and that would be a DNA guanine nucleotide. Or what's the one I'm missing? Thymine. We could have thymine on there as nitrogenous base, and this would be a DNA thymine nucleotide. But with RNA, you would have ribose as the base. It's the same adenine, but we now know it's an RNA nucleotide for adenine as opposed to this because the base pairs have changed. Notice the A hasn't changed, the phosphate group hasn't changed. The only thing changing between DNA and RNA is that base. We could have cysteine on this one, but now it's an RNA nucleotide. It's a ribosome. Ribose, so it's an RNA cysteine nucleotide. Or we could put guanine on there. But now this would be an RNA guanine or this one, which we could tell the difference because it only appears in RNA, but it's still, we know it's an RNA molecule, ribose, with a uracil, so this would be an RNA uracil nucleotide. So the only thing that changes between RNA and DNA always is the 5-carbon monosaccharide. The thing that changes between the nucleotides is the nitrogenous base. So the nucleotides can be DNA nucleotides or they can be RNA nucleotides. So the other thing that we talk about with these molecules is their function. So this is the molecule that makes, this is what RNA is, what ribosomes are composed of. That's where the ribosome comes from. Notice ribosome. So therefore, their function of these molecules is to create proteins. So remember, ribosomes are made in the nucleolus. They are the nucleolus. We put together RNA molecules to make that ribosome, which will then make the proteins. DNA, this is going to be what the molecule, that chromatin and chromosomes are made of. This means that they hold genes, and genes are instructions for making proteins. So the function of DNA is this has the instructions for making everything you need to make you live. Ribosomes will then take those instructions and actually make the things that allow you to live. Now there's one more molecule that we need to know from this section. It is not a polymer, it is a modified nucleotide. So ATP, this is an energy molecule. And we'll talk a lot about it in a couple of chapters. This is your functional energy. So when you eat food, that food can't actually do anything. But that food contains energy that we can transform into ATP. ATP is now a molecule that your body can use in order to actually do work. So when we say it's a modified nucleotide, it's going to have a 5-carbon monosaccharide as its base. And then it's going to be ribose. It's going to have a nitrogenous base. In particular, it's going to have adenine. ATP stands for adenosine triphosphate. So it's going to have the nitrogenous base here. Now, if this were a regular nucleotide, you would just have one phosphate group. But we just mentioned this is triphosphate. So what we're going to do is we're going to take that basic molecule of a nucleotide and we're going to modify it by adding 2 more phosphates. So ATP stands for adenosine triphosphate. So, and it is a modified nucleotide. So instead of 1 phosphate, we're going to have 2.